Okay, good morning. Good morning. So this morning, we'll summarize the work that's been done on the cell biology of Leishmaniasis. And by the way, this is not an area that I work in. It's an area that Dr. Saha is one of the world's experts, so he should come up and give this talk. But he is here as a resource if we have questions. Yeah. So Leishmania is an intramacrophage parasite, so a lot of work's been done trying to understand how Leishmania promastogood stages and amastogood stages gain entry into macrophages, the receptors that are used to engulf these stages of Leishmania, the nature of the endocytic compartments that they reside in, how they remodel endocytic compartments. Do they remodel them so as to delay or prevent fusion with late endocytic or lysosomal compartments? So just to remind you that Leishmania is one of the few intramacrophage parasites that ends up in a phagolysosome. Most other intracellular bacteria, fungi, parasites, they once they're taken in and are sitting in a parasitophorous vacuole or a phagosome, they have evolved interesting strategies to evade the maturation of those endocytic compartments to prevent, in some cases, to prevent fusion with lysosomes, which contain, of course, powerful hydrolytic enzymes which can kill most intracellular microorganisms. Or they have evolved strategies to escape from these endocytic compartments into the cytoplasm. So Leishmania is one of the few successful intramacrophage parasites that the replicating stages, these amastogods here, really are ultimately found in a very mature endocytic compartment in which the phagosome has fused with many lysosomal vesicles, so they are exposed to the very heart of the defensive machinery of these phagocytes. But they manage to survive largely because of this family of phosphoglycan-containing molecules on the surface, these glycocalyx structures that are on the surface and that are shed from the surface of these organisms that make them resistant to these hydrolytic enzymes in these acidified compartments. Nonetheless, there's been a lot of work to demonstrate that at least the promastogood stages remodeled the endosomes, so at least to delay the fusion with these lysosomal or late endosomal vesicles. So in addition, phagocytosis is typically associated with activation of NADPH oxidase, which generates powerful reactive oxygen intermediates, which can damage these microbes. So they do have mechanisms to prevent the signaling pathways that generate the formation of these NADPH complexes. And in addition, once they reside in these things and have established themselves within a phagolysosome, these macrophages become refractory to immune activating cytokines like interferon gamma. So even in the context of an adaptive immune response in which T cells are localized to these infections and start secreting interferon gamma, infected macrophages become somewhat less responsive to the activating signals of interferon gamma mediated pathways. So let's consider in a bit more detail the nature of the receptors for direct Leishmania entry into host phagocytes. Again, most of the work has been done with the Leishmania promastocute stages. A lot of this work has not been done with the infective stage of promastocute, so it makes some of this literature a bit confusing. As we discussed yesterday, the surface of metacyclic promastocutes is very different in terms of these phosphoglycan structures as compared to the dividing forms of promastocutes found in the sandfly midgut. So the majority of papers in which they've looked at the engagement, the uptake of promastocute stages with macrophages in vitro haven't used clearly defined infectious metacyclic stages. So it's difficult to know how to interpret the data. Nonetheless, a large number of different phagocytic receptors on the macrophages have been identified that play a role in some of these studies. These are receptors for different cleavage fragments of complement. These parasites beautifully activate serum complement, which is an innate 20 or 30 different proteins in serum that are an innate sensing structure that complement, that's why it's called complement, adaptive immunity. And the activation of these serum protein components in this cascade of activation events can lead to the deposition of these C3 cleavage fragments, these red things that opsonize the microbe, which are then recognized by receptors for these complement cleavage fragments. These are pretty high affinity receptors and really beautifully potentiate, enhance the phagocytosis of these opsonized organisms. Remember that Leishmania promastocutes, particularly Leishmania donovani, because of the LPG, have a lot of mannose terminally exposed mannose on their surface, on these LPG structures. And so these phagocytes have mannose receptors as well. Fibronectin receptors have been identified as playing a role in the uptake of these parasites. If we get to the Leishmaniae mastocyte stages, not a lot of work has been done, but some complement receptors, fibronectin receptors, and even Fc gamma receptors. These are receptors for the constant regions of antibody molecules. And it has been reported that individuals have natural antibodies, even prior to any exposure to Leishmania. They have low levels of natural antibodies that can react with some Leishmania strains. And if they're coated with these antibodies, they can, these Fc gamma receptors that have high affinity for these Fc domains of the antibody molecule, they will facilitate the uptake of these parasites as well. In a little later, we're going to talk about what we've learned when sandflies actually inoculate parasites into the skin. That in fact the entry of these parasites into the entry of metacyclic promastocute stages delivered by sandfly in the skin, their uptake by the macrophage may not be direct at all. They may be delivered by neutrophils that have been recruited to these sandfly bite sites. And it's the neutrophils that take up the parasite. And then as these neutrophils die, the dying neutrophils are taken up by these macrophages. They engulf the dying neutrophils, and because they're infected, that's how these parasites gain entry into the macrophages. So it may be an indirect method of entry. So that's a fascinating idea, which I'll show you data to suggest that's in fact how a lot of macrophages in the skin do get infected. Okay, so that's it's been a very active field. There's not a lot of agreement because people use different macrophages, different species of parasites, different stages of parasites, and not a lot of agreement which of these receptors are more important than others. We'll talk about virulence molecules that are important in the uptake and then the survival of these parasites inside of a macrophage. So we've talked a lot about LPG, which is the predominant GPI-anchored surface glycolipid on promasticoid stages. LPG, it's important to remember, when they differentiate inside of these vacuoles into amasticoids, they downregulate LPG. They don't need the LPG anymore. That tells us this impressive surface glycocalyx structure really is designed for their development survival in the sandfly vector and in the initial encounter with phagocytes in the skin. But the tissue amasticoids, they completely downregulated. So this dominant structure disappears, and you just have these Gippel structures here that are still on the surface of the amasticoids. Both stages express this GPI-anchored glycoprotein. This is the predominant glycoprotein. It's a metallo-surface protease, so it has very important protease activity. So in each case, so this is in vitro studies showing the localization of the LPG. So we're starting with metacyclic promasticates that are taken up by, I think, bone marrow-derived macrophages in vitro. The LPG is stained in red. The green shows the lysosomes that have fused with the phagosome. This is the nucleus of the macrophage. I guess these are parasite nuclei here. Anyhow, you can show this beautiful redistribution of the LPG, which has been released or shed from the surface of the promasticoid and is found in the phagosomal membranes of these phagosomes that have taken up the parasite. And that happens very quickly. So at least for the promasticoid stages, there's a beautiful transfer of the LPG molecule from the surface of the parasite to the phagosome membrane itself. And there's some pretty nice studies that demonstrate that the LPG, when it gets on the phagosome membrane, or the plasma membrane, or I guess the phagosome membrane, it prevents the translocation of protein kinase C. And so inhibiting this translocation inhibits the activation of protein kinase C. And these are, I'm going to show you a series of very nice reviews, again from Dr. Saha's review article, which I think I assigned for you. So this inhibition of PKC translocation in the membrane prevents the ability of this kinase to phosphorylate these two subunits of the NADPH oxidase P47 and P67. It prevents that phosphorylation. So it prevents the formation of the NADPH complex and their translocation to the phagosome membrane. And of course, NADPH oxidase is required for the generation of these reactive oxygen intermediates, which are typically lethal to microbes inside of the phagosome. Okay. Further, this translocation of the PK protein kinase C is required to phosphorylate these marks, and that phosphorylation of marks is involved in actin turnover and phagosome maturation and lysosome fusion. So the presence of this LPG that prevents PKC translocation will also inhibit, at least delay the fusion of lysosomes with phagosomes. But ultimately, the amastogote stages are found replicating inside of mature acidified phagolysosome compartments. GP63, that's surface metalloprotease, GPI-anchored glycoprotein, which is a metalloprotease. It is also found redistributed, it is released by exosomes and cleaves multiple host proteins, including protein tyrosine phosphatases, and these phosphatases dephosphorylate a number of substrates of the various kinases in the cell. And so leads to downregulation of pro-inflammatory responses and downregulation of the ability of the cell to present antigen. It's interesting that GP63, again, this is a phagosomal parasite, but the GP63 somehow finds its way to become perinuclear, surrounding the nucleus of the macrophage itself. So both LPG and GP63 are at least two of the probably many virulence factors that are used by these parasites to gain entry into and then survive in these phagolysosomal compartments of host macrophages. This summarizes a lot of work done by Dr. Saha's group showing that that CD40 receptor ligation, which is a critical, this signaling pathway through CD40 is a well-described critical signaling pathway for the production of IL-12, which we talked about is the key instructive cytokine produced by these innate cells leading to Th1 generation. So IL-12 will instruct NK cells and T cells to produce interferon gamma, which is, of course, the key immune activating cytokine to activate these infected macrophages for killing. And CD40 ligation is critical through P38 MAPK. But what their group has found is that I guess it's the strength of receptor ligation, is that correct? Or the amount of cross-linking of CD40 is, can lead to a really interesting dichotomy in the signaling events, CD40 signaling events, and as a consequence of Leishmania infection, CD40 ligation, that signaling is redirected along this ERK1-2 pathway, which leads not to IL-12 production, but IL-10 upregulation. And IL-10 is, of course, a powerful anti-inflammatory cytokine and prevents IL-12 production and prevents interferon gamma activation of these cells. So Leishmania infection downregulates CD40-induced P38 MAPK phosphorylation, enhances the capability of this receptor to signal along this pathway to upregulate IL-10. And finally, a lot of work's been done to show that once you've established infection in host macrophages, and even if there is a Th1 response in that localized environment, and interferon gamma, and this cell is exposed to interferon gamma, these infected cells will upregulate these host phosphatases, these host cell phosphatases like SHP1, which interfere with or inhibit this JAK-STAT1 signaling pathway, which is the key signal transduction pathway involved in interferon gamma-mediated upregulation of INOS, which is the key inducible NO synthase leading to NO generation and killing of these intracellular parasites. So all this work is difficult to put into any particular context because, again, so many different kinds of host phagocytes are used, so many different kinds of parasites and stages, and in the context of an innate response prior to an adaptive response in which interferon gamma will be present or not. But all of them suggest that the parasite collectively has adapted a huge number of different strategies to get into and then survive for a long time in these host cell macrophages. Okay. That said, there is ultimately pretty strong immunity induced by Leishmania infections. Most lesions, cutaneous lesions, will heal even without drugs. So immunity does kick in. So all of these virulence factors, all of these remodeling, the remodeling that we see inside of these host macrophages, it may delay the onset of immune response and these killing mechanisms. But ultimately, the host response does succeed in controlling these infections. So keep that in mind. In some occasions, like progressive visceral leishmaniasis in people, it clearly fails. But the vast majority of exposures to Leishmania infections, these infections are ultimately controlled. Just a couple of comments about once they establish themselves inside of these phagosomes, these parasitophorous vacuoles, these amastogotes, in order to survive and propagate, they have to acquire nutrients. So the amastogote parasitophorous vacuole remains a highly interactive dynamic organelle in order to acquire nutrients and additional membrane from the host cell to allow for parasite replication. Every time this parasite divides, it's enclosed in a closely opposed new membrane. So if you have 100 parasites, in one cell as a consequence of division, each one is enclosed within its own membrane. So it has to get membrane from somewhere. That's a lot of membrane. So it seems to get it from the ER, from the endoplasmic reticulum. Okay. And when they're inside of this vacuole, these amastogotes have to acquire nutrients and cations and carbon sources, of course. And they're delivered to the phagolysosome via either the endocytic pathway or directly from the macrophage cytosol. So this is the cytosol of the macrophage, and this is a very comprehensive review by one of the leading investigators who looks at the metabolome of Leishmania inside of these macrophages. So there's a lot going on here, but it's really to emphasize all of these transporters or symporters that are involved in the transport of purines, because these amastogotes must scavenge, as we discussed the other day, all of their purines. and many vitamins, and at least 10 essential amino acids from the macrophage phagolysosome, okay? And the uptake of these essential metabolites facilitated by these polytopic membrane transporters, which are often proton symporters, because this is an acidified vacuole that these parasites are sitting in. And so this enables the high affinity uptake of all of these substrates in the acidic phagolysosome. Okay, so there's not many investigators who are looking at the metabolism of these parasites inside of these vacuoles. So clearly there's a lot going on for them to meet their nutrient requirements. Just a word about iron efflux and influx. So these parasites have an absolute requirement for iron. Where do they get it? And the host cell has a requirement for iron. So Leishmania upregulates this molecule called hepcidin. And hepcidin induces ferroportin degradation. And this importer, ferroportin, leads to ferrous iron accumulation, and which favors parasite replication. So it degrades the efflux of iron from the host cell. So Leishmania, clever enough to upregulate this homeostatic control, hepcidin, which degrades this critical ferroportin that's involved in iron efflux. And so you get accumulation of ferrous iron in the cytoplasm of the host cell, and then the amasticotes themselves have their own transporters for iron, LIT1 and LFR1, which allow them to uptake this ferrous iron into the cell for their own needs. So just an overview. So Leishmania are phagocytized by the macrophage via a range of receptors, and the receptor choice affects phagosome biogenesis. Depending on what receptors they use, the phagosomes will look quite different. Leishmania phagosome maturation modified by mechanical action of the flagellum and a restricted fusion with vesicles at the endosomal pathway. Phagosomal pH and iron availability trigger promasticate to amasticate differentiation. In terms of intracellular growth, it depends on the interaction with ER-derived vesicles, presumably as a source of nutrients and additional membrane for the parasitophorous vacuoles. Leishmania acquires nutrients from vesicles of the endolysosomal and ER pathways, possibly lipid bodies, following their fusion with the parasitophorous vacuole. Leishmania acquires iron through heme and ferrous iron transporters. The parasite upregulates iron uptake and retention by the macrophage to enhance iron availability. In terms of the Leishmania evasion of host defenses in these cells, they upregulate host protein tyrosine phosphatases, which leads to suppressed inflammation. They interfere with JAK-STAT pathways, preventing macrophage activation by interferon gamma. They inhibit ROS generation, and this is really important, by preventing NADPH oxidase assembly. They inhibit MHC class II expression and modulate the phagosome proteolytic function to suppress substrate production for efficient antigen presentation. So they've evolved so many different strategies to survive in and persist in these innate cells. So now I'm going to show a series of slides that look more carefully at the nature of the cells that these parasites actually interact with at the site of a sandfly bite. So again, most of the studies I've just described to you are just in vitro models of how people imagined these parasites interact with macrophages. And there was no particular care taken in terms of what kind of macrophages to use. In most cases, they were just bone marrow-derived macrophages or peritoneal macrophages from a mouse. In some cases, some human cells. But particularly when we consider these very initial interactions of the insect stages that are transmitted to the skin and that initiate infection in the skin, what sorts of cells actually do interact with these parasites at the site of delivery by a sandfly bite. So these are just the possibilities of the different phagocytes in the skin. So there are dermal macrophages in the skin. There are a few monocytes. So, and we're the second lecture this morning, we're going to talk extensively about the nature of these tissue-resonant dermal macrophages that are not derived from blood monocytes, but are that are actually embryonic derived and are sitting in the skin and poised to encounter these parasites at the initial bite site. There are dermal dendritic cells, and there are neutrophils that will swarm into these bite sites as a consequence of the tissue injury, and we're going to look a lot at that. And there are not a lot of monocytes sitting in naive skin. So monocytes can eventually, once they're recruited to these inflamed tissue, they will be recruited from blood and differentiate into macrophages. But at the time of this initial exposure, there's not a lot of monocytes sitting there waiting to encounter these parasites. Okay, so you know, we're fortunate to have sandflies to initiate infections in our program, so that we can actually begin to address what these encounters at the site of parasite delivery by a sandfly bite actually look like. I think we're gonna. Well, well, let's go back to this model. So we infect, we get the flies infected by artificially feeding them through a membrane on mouse blood. So we add L major A mastigates, the tissue stage that would normally be taken up by a fly when it feeds on an infected host. So we're doing it artificially through a chick membrane. So the flies are infected by seeding mouse blood with L major A mastigates and then infecting artificially through a membrane. We wait about 2 weeks for these infections in the fly midgut to mature so that they generate these huge numbers of metacyclics in the anterior midgut. And then we take these infected flies and we'll add a few of them, 4 or 5, to these vials, which are opened on top and covered with a nylon mesh so the flies can't get out. And then we flatten the mouse ear on top of this vial containing these infected sandflies, and then expose the mouse ear to the bites of these flies for an hour or so. And we've starved the flies to make sure that they're hungry for blood. So that's what these bite sites look like after these exposures to these infected fly bites, okay? And if just by EM you can, so here's a single sandfly bite, you can appreciate the kinds of tissue damage that's been produced by a fly bite. This is the epidermis here. This is the dermal layer here. I want to stress that the sandfly biting parts, the proboscis, are very short. So unlike mosquitoes, which have very long biting parts and can probe into the skin, and their proboscis is, once they find a capillary in the skin, can actually cannulate it, can actually insert into the vessel wall. Phlebotomine sandflies, their biting parts are very short, and so they're called pool feeders. They're probing for these capillary loops in the skin, but they can't insert their proboscis to suck the blood. Their biting parts will actually just chomp away at it and produce a wound and produce a pool of blood, and so they're pool feeders. Then they'll just suck from this pool of blood. And so they do salivate, and the saliva is critical to keep the blood from clotting during this feeding event. Okay. So they're blood-sucking diptera like mosquitoes, but the way they bite is very different, and that's critical to the nature of the tissue damage you see and to the kinds of inflammatory reactions that are provoked and that these parasites will encounter when they're deposited in the skin. So here's one of these pools of blood, and here's a cross-section of a metacyclic promasticate that's been delivered to the skin. So it's sitting in this pool of blood. And that's important to remember because they're exposed to blood and exposed to serum components like complement. So their initial interactions with phagocytes will occur in this context. And as it turns out, these parasites powerfully activate the complement system and become powerfully opsonized with C3 cleavage fragments, which phagocytes hone in on and efficiently take up these parasites via those these C3 cleavage fragments. So modeling these encounters in vitro, if it's not done in the presence of blood or fresh serum, you're missing a critical component of how these parasites would normally encounter phagocytes. Okay? So we use, in order to see these parasites by the intravital imaging I'm going to show you, we use a parasite that has been stably transfected with PK as neo-DS red, so it stably expresses red fluorescent protein. So this is a beautifully infected, after 2 weeks, these infections which started down here have now migrated to the anterior gut. So we can see quite visually, you don't even need UV light. You'll see this intense red fluorescence in the anterior parts of one of these infected flies. And then we're going to do intravital imaging by 2-photon microscopy. So I'm not going to explain the physics because I don't really understand the physics, but it's intravital imaging. This is live tissue. We don't kill the mouse. The mouse is anesthetized and kept completely quiet. It doesn't move at all for many hours to do this imaging in the mouse skin, so the exposed ear is placed on this platform. And so the other important tool, you have to use a reporter mouse, and most of the imaging I'm going to show you, the reporter mouse, these are mice, these are lysozyme M GFP mice. So these bright cells you're going to see, so lysozyme, the GFP is under the control of the lysozyme M promoter, so these bright cells are neutrophils, okay? And the dimmer cells are monocytes and macrophages. Again, you're not going to see anything unless you have a reporter mouse in which certain cells are fluorescent. So the bright cells you're going to see are neutrophils. these white cells you see here, we figured out how to stain and how to stain these dermal macrophages, these dermal resonant macrophages. So these are tissue resonant macrophages that are sitting in the skin of a naive mice. These are the white cells. The blue are blood vessels. The red are some leishmania. So we're looking 2 hours post sandfly challenge. You can see some red parasites have been deposited here. And these green cells that are about to come in are the neutrophils, okay? So there seems to be three sites of where these neutrophils are swarming. These are either 3 separate bite sites from 3 different flies, or the same fly that has stuck its proboscis in, one's pulled out and put it in again. And if there's any tissue damage at all, any dead cells, then neutrophils will sense this and be recruited from the blood and swarm around to these bite sites. Again, they're near, but not exactly where these parasites are. So which is interesting. The parasites we see are mainly here. The main bite sites are there. So this fly, which was probably heavily infected, was having trouble feeding, and was trying to unblock this plug of parasites so it could get blood, and regurgitated some parasites here, pulled out its proboscis, inserted again, and maybe finally found blood in one of these sites. Okay. So this is a static image of one of these neutrophil plugs. This is just 60 minutes post-bite. So this bite site is so beautifully defined by this massive neutrophils that has swarmed into this site. And you could see where some of the parasites have been deposited. Some are in the most superficial aspect of the skin. Some are deposited more deeply in this tissue. Okay. Here's another example. Again, bite sites or probing sites by the same fly or different flies. In each case, forming these plugs, these neutrophil plugs. So it's not surprising that neutrophils are swarming into these sites. That's what they do. They're an absolutely essential component of a wound healing response. And they're essential to sterilize these wounds. That's what neutrophils do. So you can appreciate that these parasites that neutrophils are a pretty major and important cell that they're encountering at these bite sites. This is a cool intravital image of the formation of one of these plugs. This is a top view. These are the side views. I'd focus your attention here. So this is a vessel, and we're looping this. So the neutrophils are coming to arrest here, and then they undergo whatever that word is. They extravasate from the vessel and fill this space left by the biting parts of the fly. And these bite sites look the same whether the fly is infected or not. We don't see any difference. so in all these transmissions to the mouse here by infected flies, most flies deliver very few parasites. Occasionally, you find a fly that has delivered a huge number of parasites. Again, probably because of this blocked fly process in which they regurgitate. They try and dislodge this massive parasites in the anterior gut so that they can take in blood. And in doing so, occasionally a fly will deliver a huge number of parasites to the skin. So it's not surprising neutrophil gets to these bite sites. The question is, do they take up Leishmania, these Leishmania metacyclic promastogotes in the skin? And it looks like it looks like at least some of them do. Some of these parasites are taken up in this by neutrophils. So here, here, and if we rotate these images, you can appreciate that that at least some neutrophils have taken up these parasites. So it's not clear that every that every parasite ends up inside of a neutrophil when it's delivered to the skin. I think we're convinced now that if parasites are deposited some distance from the actual bite where the fly succeeds in getting blood, those parasites end up more in these tissue-resonant dermal macrophages. But parasites that are close to, proximal to these sites where neutrophils swarm into to the bite, most of them end up inside of neutrophils. Okay. It was difficult to obtain enough infected neutrophils from these sandfly bite sites, infected sandfly bite sites. So the next question is, so the neutrophils that take up these parasites, what happens to them? Are they killed, or do they survive? And then what is the fate of the parasite inside the neutrophils? What's the fate of those infected neutrophils? And we found that we could inoculate into the mouse ear, into the mouse skin, high doses of these red parasites by needle. So we can reproducibly just put a lot of parasites in the skin. There is some tissue damage from the needle. It's not nearly so localized as the fly bite site. But as you'll see, just 30 minutes after putting lots of red promastogotes, these are metacyclics, in the skin, you'll begin to see a lot of neutrophils come in, and they track, they migrate directly to where these parasites are sitting in the skin. And so almost all of the parasites, again, these are needle injections, not sandfly bite sites. But it suggests, and you'll notice, you know, this is just 30 minutes after putting in these highly motile promastogotes, metacyclic promastogotes. None of them are, well, that one's moving a bit, but they become pretty stationary. So something stabilizes them, prevents their motility. They're attaching to something in this tissue. And then they seem to be attracting the neutrophils. They're saying, come and get us. They're not doing anything to evade this process of neutrophil infiltration and neutrophil phagocytosis of these cells. It's as if they want to be eaten by these neutrophils. And as you'll see, that's because this is an advantage to them. It's part of the infectious process. So because the neutrophils from these reporter mice are green, and the parasites are red, we can use a cell sorter to purify infected neutrophils from the skin, so you can get this beautifully homogeneous population of infected neutrophils from the skin. So virtually all of them have taken up parasites. If we do EM, the viability is clear. So this is a metacyclic promastogote that already looks like it's an amastogote after being taken up by these neutrophils. So it's sitting in a vacuole in these neutrophils. There's no evidence that that this parasite has been damaged at all. And the proof of that was to put these infected neutrophils from these sorts into promastogote growth medium. And 96% of the if we put one infected neutrophil per well, 96% of them will grow out promastogotes, the insect stage. So these neutrophils so efficiently take up these parasites, and they don't kill them. This is a nice series of a nice sequence of imaging, static images, looking at the same bite site, the same parasite delivered by the bite of one sandfly. And here's the neutrophils that have swarmed into that bite site at 2 hours. You can't see the red parasite there, but there's just one or two. Here it's 4 days. And it's the same mouse, the same live mouse. And using the hair follicles as sort of signposts to get back to the exact same bite site on these for over 150 days, you could follow the growth of these, the growth of this single parasite delivered by this one sandfly. So by 12 days, there's this clear growth of parasites. There's still a lot of neutrophils about, but many other cells have come in here. And at the time you begin to see the actual formation of a lesion, it takes about 1 month. So now there's a mass of parasites here, and still just a huge mass of parasites at 43 days. And it's not till 2 months that the immune system has begun to kick in, and now you've begun to kill and clear out these parasites. So the point here is these parasites are growing unencumbered by an immune response for weeks and weeks and weeks. You know, you might appreciate that the immune system is going to have a hard time sensing just a few number of parasitized cells here. But by, you know, certainly by 2 weeks or at one month, they should have been able to sense this massive ongoing infection in the skin. And yet they continue to grow for probably 2 months before the immune system is kicked in. And then once it's kicked in, you can see how efficient it is at killing these parasites. We wanted to know if these early events involving neutrophils contributed to this remarkable efficiency of these parasites to establish infection in these first days and weeks after delivery by sandfly bite. So we took naive mice, these are black 6 mice, and we treated them prior to sandfly biting with an antibody, this RB6 antibody, which depletes neutrophils. This is how we stain neutrophils in tissue from the mouse ear dermis. This is the population of neutrophils. When we treat these mice just for 1 day, it's a powerful depleting antibody. We see no neutrophils in these bite sites 1 day after sandfly biting. So now we've initiated these infections in the absence of these neutrophils that swarm into these bite sites in the first hours and days after transmission. And when you do that and you look at the numbers of parasites in the skin. So these are, each one of these circles is one ear from one mouse. And these are the parasite loads. So the first thing to appreciate is when we deliver infections by needle, you control the inoculum size, and these recovery of parasites, you would only need to do 5 mice because they would all look pretty much the same. But that's not how the real biology works. These sandflies deliver hugely different numbers of parasites to the skin. So if we're just looking 4 weeks in these control mice that we haven't depleted the neutrophils, some of these mice have over 1 million parasites in the skin. Some have already cleared out the infections. So that's why we had to use so many mice, because the real biology is associated with these fantastically different ranges of transmitted doses of parasites. So that's how it really happens. And it may be that the reason some people develop leishmaniasis and others don't after exposure to an infected fly is just dose. Some flies have delivered a huge dose to the skin, and others have delivered a really small dose, right? Okay, but what happens when in these mice that have been transmitted infection, we've gotten rid of these neutrophils at the bite site? So a whole lot more of these mice have cleared out their infections by 4 weeks after transmission by bite. So, and some haven't, but a whole lot more have, suggesting neutrophils really do promote the infectious process. The parasite seems to be exploiting this acute neutrophilic wound healing response to the bite to establish infection in the skin. So how do they work? Why do they promote this infectious process? So before we started this work, actually doing the intravital imaging of sandfly bitten ears, a group in Germany, Van Zandbergen and his group, they had already proposed, based on some in vitro studies, that neutrophils will take up leishmania, and they don't kill them, which is what we saw, and that those neutrophils, at least in these co-cultures with macrophages or dendritic cells, the neutrophils are short-lived cells, and they will begin to die. And in fact, their uptake of parasites promotes their apoptotic program, so they die quicker. And then if you cold culture with macrophages or dendritic cells, these phagocytes will clear out, will capture these dying neutrophils. And they call this the Trojan horse model. It's a way for these parasites to gain entry into the macrophages, not by their direct interaction and uptake, by the receptors and ligands we talked about at the outset, but again, indirectly via this Trojan horse model inside of a neutrophil. And the advantage to the parasite is the receptors for dying cells are not activating receptors. Because these kinds of phagocytes, one of their functions homeostatically, cells are dying by the billions every day. And so they have to, dying cells have to be cleared out homeostatically. And the last thing you want to happen when you're just clearing out and recycling the contents of dying cells is that to provoke inflammation. We'd be dead instantly if that happened. So the receptors for the clearance of dying cells are deactivating receptors. They're powerfully deactivating receptors. So they deactivate these pro-inflammatory activation programs. They deactivate phagosome maturation, and they lead to the production of these anti-inflammatory cytokines like TGF-beta and IL-10. Okay. Again, these are normal homeostatic processes. Cells are dying under normal steady-state conditions all the time, and they have to be cleared out quietly. So being inside a dying neutrophil these parasites can gain entry into a host cell macrophage, which is where they ultimately want to be to replicate. So these are some recent images based on the fact that only recently have we been able to stain these tissue-resonant dermal macrophages. These are these blue cells. And you sort of have to use your imagination, look carefully, but we think it demonstrates these infected neutrophils that are being captured and eaten by these Dermis macrophages. Let's see, here's an infected, whoop, see it disappeared. So we'll loop it. See, it's carrying a parasite and now, okay. How about this one? Or this one. So we think when these neutrophils come in and they associate with these dermal macrophages, and then they disappear, it's because they're being engulfed by these dermal macrophages. Which is sort of cool. This sort of thing hadn't been captured before. Whoops. Okay. So some other images. Let's follow this one. I don't know if it's delivering a parasite. It's clearly being eaten. No, I think it is delivering a parasite. So about this one here. Yeah, this one here. Watch this one. Okay, yeah. I think that one was pretty evident. It starts here. So it's clearly delivering a parasite to this. So you can appreciate how inflammation is resolved, how it goes away by the action of these phagocytes in the skin, and that is as important a function of these cells as is their function in antimicrobial defense. This homeostatic function of clearing out dead cells. Okay. And we've done some study to show that it's not just macrophages in the skin that function in this manner. It's also dendritic cells, which of course are the more important cells in terms of presenting antigen to T cells and initiating a T cell response. So if we recover the dendritic cells just 1 day after infection with these red parasites, and then we gate on just the CD11C high and MHC high cells. These are the dendritic cells, not the macrophages in the skin. And we look at the RFP positive cells. So these are infected dendritic cells, and we ask the question, well, how did they get infected? Did they take up the parasite directly, or did they capture these infected neutrophils? So we stained for myeloperoxidase, and it was only the infected cells, these RFP positive cells, that were myeloperoxidase positive, suggesting, and these dendritic cells on their own are myeloperoxidase negative. So this does suggest that they've captured, and if we treat with this neutrophil depleting antibody and do the same experiment, then the infected neutrophils in this case are myeloperoxidase negative. So in the absence of neutrophils, the dendritic cells do get infected directly, and they're myeloperoxidase negative. But normally, their uptake, their initial encounter with the parasite is in the context of parasitized neutrophils. again, that's not so surprising given the huge numbers of neutrophils that swarm into these sites, okay? So what happens to those dendritic cells that capture neutrophils, either infected or uninfected? So these are just co-cultures. These are in vitro co-culture systems using bone marrow-derived dendritic cells cultured with neutrophils that we get from infected skin. And they very efficiently, so these are some slices through confocal images showing in blue here a dendritic cell that has captured a neutrophil, this green neutrophil. That's an infected neutrophil. If you co-culture with, that's an uninfected, if you co-culture with an infected neutrophil, now here's a red parasite inside of a neutrophil, inside of a dendritic cell. And it's very easy to do this. You co-culture these cells, these dendritic cells just love to eat these dying neutrophils, because that's what they normally do. And the consequences of that in terms of dendritic cell function, that's what we're looking at here. We're simply using the dendritic cells that have captured infected or uninfected neutrophils and co-cultured them now with T cells for a transgenic CD8 T cell with specificity for a heterologous antigen, not Leishmania, just we want to look at just T cell function, soluble ovalbumin. Okay, so we're co-culturing with these CD8 T cells, and we're looking at the ability of these dendritic cells to induce the proliferation of these CD8 cells in response to ovalbumin. So this is the control. When they proliferated, they dilute the CFSE signal because each cell division results in half the amount of CFSE inside the cells. But if we use dendritic cells that have captured infected neutrophils, they don't proliferate at all and they don't secrete interferon gamma. So they're completely shut down in terms of their antigen presentation function to T cells. So again, we're talking about really early events in the skin that prevents, that allows these parasites to get inside of a macrophage without activating it and get inside of a dendritic cell without that encounter leading to presentation of antigen and priming of T cells. So homeostasis and clearance of apoptotic cells under steady state conditions, the capture of apoptotic cells by dendritic cells has been shown to suppress dendritic cell maturation and is a crucial homeostatic process to maintain peripheral tolerance. So Ralph Steinman, the guy who won the Nobel Prize for the discovery of dendritic cells, his work in the latter years of his life was to look at the function of this sort of process in maintaining peripheral tolerance. This constant capture of dying cells in normal steady state conditions is what suppressed DC function and helped to maintain peripheral tolerance. In inflammatory settings, the clearance of apoptotic cells, like these neutrophils that are swarming to this bite site, by macrophages and DCs is central to the resolution of inflammation. So that's how this inflammatory response to these bites, that's how you resolve all that inflammation. And this process, this capture of dying cells as a homeostatic process is so critical that there are redundant receptors and ligands involved in the recognition of dying cells and in the various molecules that dying cells present to phagocytes for them to be cleared, sensed early and cleared efficiently. So it's an incredibly active, incredibly complicated field. But this is just so that you can appreciate the redundancy of this process, which tells us how absolutely critical it is in homeostasis. And so this is just what the parasite is taking advantage of. So inhibition of innate responses by Leishmania in dermal sites of Leishmania transmission by sandfly bite, the antigen presentation functions of dendritic cells and the killing functions of macrophages become subordinate to their role in the clearance of dying neutrophils, such that their immune functions are delayed until the acute inflammatory response is resolved. So eventually these killing functions kick in, these antigen presentation functions kick in, but you saw that it takes weeks and weeks. And by that time, the parasite has succeeded in doing what it needs to do, is persisting long enough to be picked up by another sandfly and transmitted to another host to maintain transmission cycle. Some of the virulence factors, LPG and GP63, clearly inhibit some activation pathways in infected cells. This last point, these same conditions will operate in sites of challenge exposure to compromise the expression of immunity in vaccinated hosts. So when we start discussing vaccines tomorrow, this is part of the reason it's hard to get vaccines to work for these kinds of vector-borne pathogens. And then we'll just finish up a few more slides to sort of move on now to this link between innate responses and adaptive immunity. So eventually, these infections are controlled. T cells do kick in, and innate responses involving toll-like receptors, IL-1 production, do kick in, do play a role in mediating protection. So this is just a reminder of the different TLRs on innate cells. These are the surface membrane TLRs. These are the endosomal TLRs that sense nucleic acids. So a fair amount of work's been done now in Leishmaniasis mouse models. Do TLRs play a role in host resistance? So MYD88 is the adapter protein for most of these TLRs. So the blue is the mid-88. So it's only TLR3 that doesn't signal through this adapter protein. So a mouse that lacks this adapter protein, mid-88 has non-functional TLRs for most of the TLRs. And in a normally resistant mouse that lacks mid-88, they're unable to control Leishmania major infection. Okay. So that was a finding quite some time ago. So these parasites do somehow activate through toll-like receptors in innate cells. So despite all of these inhibitory pathways that they have, eventually they are signaling through TLRs. This was work done down in Brazil to show, so which TLRs are they using? And this is the best data to indicate it's these endosomal TLRs, TLR 3, 7, and 9. So this is a mouse. This 3D mouse is deficient, genetically deficient in all of the endosomal TLRs, which again look at nucleic acids. So somehow the parasite DNA is accessing these endosomal TLRs, and in their absence, the infection is much worse. Okay, there's some published data that LPGs activate through TLR2 and TLR4. But again, keep in mind that LPG is really not on amastigote forms. So the exposure of LPG to the immune system, these innate cells, will be ever so slight. It's just the few metacyclics deposited in the skin. And then the LPGs, again, are not expressed by amastigote forms. So these endosomal TLRs are somehow sensing DNAs from the amastigote tissue forms, and they're required for host resistance. So they upregulate pro-inflammatory cytokines like TNF, like IL-6, and especially pro-IL-1 beta. And IL-1 is such a critical central mediator of inflammation and immunity, but it doesn't work until it is processed by caspase-1, which requires the assembly of this cytosolic inflammasome, this macromolecular complex that we talked about, the inflammasome, which leads to the activation of caspase-1, which cleaves pro-IL-1 beta, leading to the release of the mature form of IL-1 beta, or IL-18. But there's not a lot of evidence IL-18 is playing such a strong role in these infections, but IL-1 for sure. Again, this is the steps in the activation of the NLRP3 inflammasome, but leading to this cytosolic macromolecular complex, the active form of caspase-1 that cleaves the proform of IL-1 beta and IL-18. So this was, again, work done down in Brazil using mice deficient in various components of the inflammasome. The ASC, or caspase-1 itself, or NLRP3, that's the nod-like receptor. So it's the NLRP3 inflammasome. So the parasites trigger through TLRs, and but we think the critical TLRs are these endosomal TLRs. The DNA and the RNA from the parasite trigger these endosomal TLRs, which upregulate pro-IL-1 beta. And then the triggering of this cytosolic sensor in this NLRP3 inflammasome is still not understood. Is it directly from parasite components or some change in the physiology of the cell, such as ROS production, so recombinant oxygen species, or potassium efflux? Both of these things are known to activate these nod-like receptors. But whatever these events are, it does lead to the activation of this inflammasome and maturation and release of the active form of IL-1 beta. So I think, yeah, I think let's take a break. And let's take questions first, and then we'll take a break. So there's a lot going on in these encounters in terms of what phagocytes to look at, what parasites to use, what readouts to look at. I think the overall take-home message is these initial counters clearly favor the initial establishment of the infection, whether it's indirectly through engulfment of parasitized neutrophils or directly through these virulence factors that deactivate various activation programs in host macrophages. Those initial encounters clearly favor the early establishment of infection for days, or it looks like weeks before the immune response kicks in. And at least some of those immune responses are linked to these innate responses that ultimately can occur in these innate cells that direct, however slowly at first, the priming of T cells and the control of these infections. So it takes a while, but eventually the host, in most cases, can do what it needs to do to control these infections.
So you have mentioned about, you did not mention about the CD8 plus cells, T cells, because the CD8 plus T cells can in turn secrete interferon gamma, which can activate macrophages for their further action. So,
so that's true. We haven't really mentioned the effector functions of CD8. We're still on, today we're mainly still talking about innate responses. So tomorrow we'll start talking about the adaptive immune responses that are required to control these. So tomorrow we'll focus more on the T cells that are activated by these innate responses. And I'll include some discussion of CD8 cells. Although we'll mainly discuss CD4s, because that's still the most important cell for controlling these phagosomal pathogens. Okay.
Sir, I have also read that the CD8 plus cells secreting INF, interferon gamma, can also inhibit the TH2 response towards the infection.
Yeah, I mean, it's not so clear how gamma will directly inhibit those pathways. I mean, I think that's probably true at the level of innate cells that have been acted upon by interferon gamma. So the problem is we don't really understand at the innate cell level what drives Th2 development. We don't really even know where IL-4 comes from. Okay. We know that innate cells, we know the instructional program for Th1 development. It's IL-12 from mainly dendritic cells. But we really don't understand why Th2 responses come to predominate a response. Certainly in terms of IL-4 and the sources of IL-4. And one idea is it's just a default pathway. In the absence of the appropriate signals for IL-12 development, these innate cells will then direct T cells along a Th2 development pathway. I suspect that's not true, but yeah.
Investigators reinfect the other phagocytic cells. Any other mechanism?
You're talking amastigotes? Yeah. Do they infect other phagocytic cells? Well, I think that's true. So, what sort of other phagocytic cells are you talking about? Are you talking about neutrophils or dendritic? Yeah, I mean, any phagocyte that encounters an amastigote, I am certain, in these tissue environments will take them up. I mean, that's what phagocytes do. So you just have to keep in mind, in these inflammatory environments, what are the main cells that are there? So there's going to be tissue macrophages, and there are going to be T cells that are influencing other cells, recruiting other cells because of the chemokines that are produced in these sites. So monocytes will be coming in, which will differentiate into macrophages. We see neutrophils in these inflammatory sites. So all of these phagocytes, if they encounter an amastigote that is released from an infected cell, they'll take them up. But the predominant cells that are continually recruited to these inflammatory lesions are monocyte-derived cells. Those are the predominant new phagocytes coming in. There are neutrophils that constantly come in, but this massive number of neutrophils, they're mainly associated with these early bite sites. And then the neutrophils are cleared, but there's always some neutrophils in these lesions. And they're infected. You'll see them infected. But the parasite grows best in macrophages. Okay?
Why we are not targeting neutrophils or as a vaccine? Because complement 3 will they capture the parasites and store for a few hours for another phagocytic, prevent from another phagocytic cells. And why we are not targeted to
Yeah.
prevent
Well, that's an interesting idea. So how would that work? To target neutrophils, you would have to do that very transiently. You wouldn't want some vaccine that made that host unable to make a neutrophilic response to infection or a wound. You'd like to be able to, if you could know when an infected fly was about to bite an individual, you would love to be able to deplete neutrophils at that time, but you would have no idea when that is, right? So we can do that in the laboratory because we know when we're going to expose the mice to these infected flies. Otherwise, it would be a very dangerous thing. I mean, neutrophils are such an essential component of a wound healing response that you wouldn't want to compromise a neutrophilic response over the long term. Yeah. Other questions? We'll show a few more of these videos in the next talk. Okay. Okay. So now we're going to continue to focus attention on the nature of the cells in the skin, where all Leishmania infections are initiated, whether they're producing localized cutaneous lesions or whether they disseminate and produce visceral leishmaniasis. Again, all infections are initiated in the skin when sandflies deliver promastigates, metacyclic promastigates, to the bite site. And I'm gonna discuss now these tissue resonant macrophages that are in the skin. Previously, most people have thought that infections involve bone marrow derived macrophages, or in other words, macrophages derived from blood monocytes that are recruited to these inflammatory sites. So we're going to discuss another population of macrophages that really had not been well studied before, and it's termed a Dermis resonant macrophages, tissue resonant macrophages, and how they and their function in Leishmania major infection in a mouse model. So the emerging paradigm to explain the special ontogeny of tissue-resonant macrophages. So macrophage biology has been a very active field in the past few years because it's revealed this really interesting dichotomy between macrophages that are derived from blood monocytes versus macrophages that are embryonic derived and locally maintained in different tissues. So the long-held belief was that macrophage populations are continuously replenished by monocytes from the bone marrow. So the new understanding is that, well, there are macrophages that are replenished from bone marrow in inflammatory sites. In fact, there are tissue-resonant populations that are embryonic derived, either from fetal liver or from yolk sac, and that's in contradistinction to bone marrow derived monocytes. So if you look in the brain, microglial macrophages are virtually all embryonic derived from embryonic yolk sac. If you look in the skin, Langerhans cells, we haven't discussed Langerhans cells, these are cells in the epidermis. They're in fact derived from both yolk sac and fetal liver cells, and not from bone marrow. In the lung, alveolar macrophages are embryonic derived. In the liver, Kupffer cells are embryonic derived. They are not derived from blood monocytes, and they're not replaced by blood monocytes. So tissue-resonant macrophages in these different tissues, they're embryonically seeded from yolk sac or fetal liver, and they're self-maintained independently of adult hematopoiesis. So they're self-independently of precursor cells from the adult bone marrow, okay? And that was not understood before, so that really is a new sort of paradigm in macrophage biology. So since Leishmania is an intramacrophage parasite, it's important in the various tissues in which Leishmania can be found. Certainly the skin is where they are all found, at least at the outset. What's the role of skin-resonant macrophages in these infections? Well, this makes the point that the diversity of tissue, there's an interesting diversity of tissue-resonant macrophages in these different organs. So this is just transcriptional profiles, transcriptional signatures looking at principal component analysis of the tissue-resonant macrophages from the brain, spleen, liver, lungs, peritoneum, And they each have a different transcriptional profile. So these tissue-resonant macrophages, while they're all embryonically seated, they are conditioned by specific microenvironment in the tissues in which they're found. But all of them, so despite these individual transcriptional profiles, they all have some commonalities, and they're generally classified as M2-like. So we haven't really discussed M1 and M2 macrophages, but we'll begin to do so now. By M2-like, these are alternatively activated macrophages. That's the general name given to macrophages that have been acted on by Th2 mediators, like IL-4 and IL-13 and IL-10. And so they're not classically activated, as are M1 macrophages acted on by TLR signals and particularly interferon gamma. M1 macrophages are, their activation program results in killing of the intra-macrophage pathogens. M2 macrophages do not kill these pathogens. So tissue-resonant macrophages, at least in steady-state conditions, they have an M2-like activation program. And part of that M2 program involves homeostatic phagocytosis of cellular material, such as apoptotic cells. And we already looked in the skin at those macrophages I was showing you in the skin that seemed to be capturing and engulfing those neutrophils that came in and were dying. That is just part of their homeostatic program. Macrophage, the function of these tissue-resonant macrophages is probably not host defense. It has a reparative function. They function in tissue repair and remodeling, in wound healing, and the resolution of inflammation. And they require signals from Th2 signals, either IL-4 and IL-13, along with capturing apoptotic cells to maintain their M2-like program in the tissue. Okay. So the role of tissue-resonant macrophages in infection-driven inflammatory settings have not been well studied, especially in the skin. So most of their function is just in steady state. What do they do either in steady state conditions or in the context of wound healing and tissue damage? But in terms of infection, particularly in the skin, they hadn't been well studied. In terms of infection. So Leishmania major is a beautiful model in the mouse in which to study the function of tissue-resonant macrophages in the skin in the context of infection. And we've seen this slide before. And we're focusing on these early events in the skin at the site of delivery of parasites by sandfly bite. And we now know that neutrophils play a pretty important role in these initial encounters with parasites in the skin. But ultimately, the parasite does have to get into macrophages to grow, to propagate inside of these macrophages. And neutrophils will deliver these parasites at least to some of these macrophages. But when we say macrophages, what kind of macrophages are we talking about? Well, we used to think we meant mainly, if not exclusively, inflammatory monocytes or inflammatory monocyte-derived dendritic cells in macrophages. Clearly, these still are important, and we'll talk about those, but we'll talk about them in the context of these tissue-resonant dermal macrophages. So these are the different populations of phagocytes that we now know these parasites might encounter in these inflammatory environments. And we'll discuss it in the context of zoonotic cutaneous leishmaniasis in the mouse model. So this mouse model, from the first lecture, you'll remember the initial studies describing CD4 subset development, describing Th1 and Th2 cells. The Leishmania major model in these different inbred mouse strains was the first in vivo model to demonstrate the functional dichotomy of different CD4 subsets. So in the B6 mouse here, they controlled the infection, and that was associated with the Th1 response and the secretion of high levels of interferon gamma, which acted upon macrophages to classically activate them along this classical M1 activation pathway for parasite killing. By contrast, the BALB/c mouse, which did not control the lesions, they made a polarized, a predominant Th2 response, high levels of IL-4, which acted upon macrophages along this alternatively activated program that supported the growth of the parasite instead of killing it. So that's the classical Leishmania infection model in the mouse. The problem is there's little evidence that non-healing severe forms of leishmaniasis in humans is associated with Th2 polarization. So we think the resistant mouse model well enough reflects the kinds of host resistance mechanisms that we see in people. But in terms of the susceptibility, this really hyper-susceptibility that you see in the BALB/c mouse that fail to control these cutaneous lesions, you just don't see that in patients with cutaneous leishmaniasis. So the analysis of chronic localized non-healing lesions in humans has in fact revealed increased expression and even high levels of inflammatory cytokines, and not a lot of IL-4, but detectable levels of IL-10, low Th2 associated cytokines. So in these people, and this is a case that was at NIH a long time ago, a chronic lesion wouldn't respond to therapy, was not self-healing, and yet this patient seemed to have an intact immune response, made high levels of T-cell responses, including interferon gamma production, and yet failed to control these lesions. When the parasite from this individual who obtained these infections from West Africa was put into a conventionally resistant B6 mouse. So this is this strain from Senegal, West Africa. It's interesting. You take it from a human, it produced chronic lesions in this human. You put it now in a conventionally resistant mouse model. This mouse also could not heal these infections. And that's in comparison to a more typical strain from the Middle East, which produced these nodules that took weeks and weeks, but eventually healed the way it does in most people. So there are non-healing strains of L major even in a resistant mouse model. And so if we look at the lesion pathology, the lesion size, this strain's non-healing. This strain from Israel heals, takes a long time, but it heals. If we introduced another parameter of this infection, this pathology score, this is just the degree of ulceration. This healing strain which produced these nodular lesions, they never really ulcerated. Whereas this non-healing strain, you can see complete erosion of the ear dermis. So this pathology score of beginning of an ulcer, ear half eroded, no ear left, that's that score. So it's a non-healing strain, produces really severe pathology. So what's going on? Are they just not making the appropriate Th1 response needed to control these infections? This is just the histopathology of the healing, non-healing strains. This looks at parasite burden. There's some control. There's, again, the healing strain takes a long time, but they eventually efficiently clear most of the parasites from the site. this non-healing strain, there is some control, but not efficient control at all. There's millions of parasites in the site. But if you look at the immune responses, and now we're looking at the T cells that have been recruited to the lesion itself, to the skin. If we look at the healing strain, these are the CD4 T cells, and the number of those CD4 T cells making interferon gamma, so the Th1 response. We find that the healing and the non-healing strain look the same. If anything, the non-healing strain has an earlier and even more powerful Th1 response, huge numbers of CD4 cells, and they also make a lot of CD8 cells making interferon gamma in the site, and yet they're not controlling these infections efficiently. So we think this is a much better model for severe disease than the BALB/C Th2 polarized response. We just don't see that in people, including people with visceral leishmaniasis, and we'll talk about that tomorrow. The immune responses in patients with full-blown visceral disease, they're not controlling infection, but in fact they have some intact Th1 responses, just like this non-healing cutaneous lesion. Okay, so as useful as that BALB/C L major model was to look at the development of Th2 cells in vivo, in an in vivo infection model, and continues to be useful to understand the factors that control Th2 development. It hasn't been terribly informative in terms of what really goes on clinically in these severe forms of leishmaniasis in people. So that's important to keep in mind. Okay, so what's the problem in these mice? So this is the work. For years we worked on this problem. We couldn't figure it out. That there's a postdoc in my lab, a Korean postdoc, Sang-On Lee, who we have some confidence has gone a long way to figuring out what the problem is in these mice, why they can't control these infections, and he decided to compare the outcome of infection of the healing and non-healing L major strain in innate cells, in bone marrow-derived macrophages in vitro. And what he found was there was just more efficient uptake of the non-healing strain, this red strain, compared to the healing strain. In these cells, they just got into these cells more efficiently, bone marrow-derived macrophages in a dish, okay? This is in vitro. Once they got inside, if you just focused the attention on the infected cells, they seemed to grow the same once they got inside these cells. So it was the initial uptake, and not just of the metacyclic promastogates, even the amastogates were more efficiently taken up by macrophages, the non-healing strain compared to the healing. But once they got inside, they grew the same. Okay. So that was the first sort of difference that we could appreciate at the level of these innate cells. And that preferential infection of bone marrow-derived macrophages by the non-healing strain was mannose and mannose receptor dependent. So I'm not going to go through all this, but if we inhibited, so there's this preferential uptake of the non-healing strain. If we threw BSA mannose into those cultures, that preferential uptake disappeared. If we use mice with that were genetically deficient in mannose receptor expression, that preferential uptake of non-healing strain disappeared. So mannose had the mannose receptor is expressed on these bone marrow-derived macrophages. So when you grow out bone marrow-derived macrophages using MCSF, you see a nice expression of the mannose receptor, okay? And in fact, if we then, we wanted to see what happens if we throw various cytokines. If we convert these bone marrow-derived macrophages into M2 macrophages using gamma and LPS, we saw a bit of a downregulation of mannose receptor expression. But most intriguingly, if we used IL-4 plus IL-10, we saw a massive increase in the level of mannose expression on these cells. And So if we converted these bone marrow-derived macrophages using these conditions into M1 or M2 cells, if we made them M1 classically activated cells, we saw now no difference in the uptake of the healing or non-healing strain. And in each case, these powerfully activated macrophages would kill the parasites that were taken up. But if we now converted these into strong M2 alternatively activated macrophages, we saw this difference in initial uptake. But again, once they got inside, they grew the same. But there's no killing by these M2 cells. Okay, so that's the in vitro stuff. We're done with the in vitro data. We're going to talk now exclusively about what's going on in the skin, in the dermis of these C57 black 6 mice. So dermis resonant macrophages had never really been described before because they hadn't been looked at. So using mannose receptor, which was so beautifully upregulated on these M2 cells in vitro, could he find a population of mannose receptor high cells on myeloid cells recovered from the skin? So this is just the steady state skin, no infection, normal skin. And these are the various markers we use to define myeloid populations. They are CD11b positive. That's the key phenotypic marker for myeloid cells in any tissue. And then we excluded, using these different antibodies, eosinophils and neutrophils. We were just interested in these myeloid macrophage populations. So we're gating on these cells now. And we're now using 2 other phenotypic markers, mannose receptor expression and LY6C, which LY6C high myeloid cells is a classical marker for inflammatory monocytes. So these P1s, LY6C high, CD11b positive cells, these would be inflammatory monocytes from blood. In normal skin, there's not a lot of them because there's nothing going on in normal skin. But there are these P2 and P3 populations are populations of myeloid cells that are mainly dendritic cells. Dendritic cells sitting in the skin, in naive skin. But the most interesting population is this P4 that's sitting in steady state dermis, mannose receptor high, LY6C intermediate. And using various other markers, we could identify these clearly as dermal macrophages. So they have these other markers. MER-TK is a very important macrophage marker. They were MAC class 2 negative. So as antigen presenting cells, they probably wouldn't work so well. So we identified this mannose receptor high population sitting in naive skin as a dermis macrophage population. P1 are inflammatory monocytes, P2, P3 are monocyte-derived dendritic cells. So these are the main populations of myeloid cells sitting in the normal skin. This is what these populations look like. This is the P4 dermal resonant macrophage population. So this slide was using various markers that have been ascribed to on other tissue resonant macrophages from other tissue like the brain, the lungs, the liver, markers that identified those tissue resonant macrophages as M2-like. So first of all, mannose receptor was one of the first phenotypic markers that identified macrophages as alternatively activated. So the important work done in Oxford, I think it was already in the 90s, when they threw IL-4 and IL-13 on bone marrow-derived macrophages, and they termed them alternatively activated M2 macrophages. They found that mannose receptor expression went way up on those cells. Okay, so already the fact that cells sitting in the skin, these macrophages sitting in the skin, are mannose receptor high, which is how we identify them, That's a strong M2 marker. And then all of these other markers, including receptor for the IL-4 receptor alpha, and all of these other receptors, which are various C-type lectins and angiogenic receptors, and IL-10 receptor. These are, if we compare the P4 dermis macrophage population with these other monocyte-derived populations, they are upregulated in steady state normal skin for these M2-like markers. So they're sitting in normal skin already as M2-like alternatively activated cells. So these dermis resonant macrophages share similar markers with tissue resonant macrophages in lung, liver, brain, adipose tissue. They're M2-like with fundamental roles in tissue homeostasis, including wound repair and the resolution of inflammation. They're not really there for host defense. They're there for these tissue reparative function during wounding or inflammation. They're there to resolve inflammation. Okay. So one of these homeostatic functions of M2-like cells is they love to capture dying cells. So we already discussed that, and we think we've captured some real-time video intravital images of tissue resonant macrophages capturing dying neutrophils in the skin. This was an experiment just taking apoptotic thymocytes and injecting them into the ear dermis, and they're CFAC labeled, and then you recover the different populations of cells from the skin, P1, P2, P3, and look for cells that have taken up these CFAC labeled dying thymocytes. And it's really only the P4 cells that take them up. So this is what they like to do, and we've already seen that in terms of dying neutrophils. But now, we're using this tool, this Manocept, to stain intravitally. It's a small compound. It's a linear glucose polymer background with these mannose moieties, and it's fluorescent, so we can inject it into a mouse, and very quickly it gets into all these tissues. And because of these mannose moieties, it binds beautifully to these mannose receptor high dermis resonant macrophages. So these are these P4 dermis macrophages are the only ones that stain brightly when we inject this Manocept, okay? So, the red is Evans blue for the blood vessel. The green cells are the Manocept for dermis resonant macrophages, and again, this is intravital imaging. The blue is the second harmonic. So the first thing you'll note is these dermis resonant macrophages tend to be perivascular. They tend to line the vessels. This is steady state skin. This is naive skin. So they're sessile cells. They're not moving. They're perivascular. They're not moving around. This is the red blood cells flushing through the vessels. But if you focus at high resolution on these cells, the cells are not moving, but their processes are moving. Their dendrites are constantly moving. They seem to be just constantly sampling tissue, sampling the surrounding tissue. So that's not a cell moving. That's a dendritic process of the cell. Okay. Okay, now we're going to address the origin of these cells. Where do they come from? So do they come from blood monocytes? Or are they embryonic derived, the way most other tissue resonant macrophages were shown to be? This hadn't been done for skin resonant macrophages, except for Langerhans cells, these cells in the epidermis, which were well described as having an embryonic origin. So the way this is addressed, these are somewhat complicated experiments, but these are bone marrow chimeras. So the mouse, the recipient mouse, is lethally irradiated. So you kill off all the dividing cells, but then you have to reconstitute the cells of hemopoietic origin with new bone marrow cells. And we use a donor that has a congenic marker, so we can recognize the adoptively transferred cells as cells of donor origin because of this marker. And if we're going to look in the spleen and the ear in infected and non-infected animals. So the donor cells are the CD45.2 marker. The marker from the mouse, that is the recipient mouse that has been lethally irradiated, is the CD45.1 marker. So after 1 week transfer, most of the cells are still of host cell recipient origin. But by 2 weeks, even in an uninfected mouse, in the spleen and in the ear, the P1, 2, 3 populations have already begun to be replaced by the donor populations of bone marrow-derived cells. And in an infected mouse at 4 weeks, almost all of the cells are of bone marrow donor origin. So the gray here, except for the P4 population. That remains of the recipient type, and even in the infected mice. So all of the other myeloid cells in the skin have been completely replaced by blood monocyte-derived cells, but not the P4 population. So it tells us it's radio-resistant. It's not dividing. If it's turning over at all, it's doing so very slowly, so it's not destroyed by this irradiation. And it is not replaced by adult bone marrow-derived cells. So this was a key to our understanding that it is not of bone marrow origin, but undoubtedly of embryonic origin. The other way to look at this is to generate these parabiotic mice. So we're taking these 2 mice that have these congenic markers, so their cells are marked by this phenotypic CD45.1 or CD45.2 congenic marker. Otherwise, the mice are identical. And the mice are sewn together. This is a conventional technique. I know it seems crazy. So after a couple of weeks sewing these mice together, they share their blood supply. And they seem viable enough, happy enough. They have to like each other because they can't get away from each other. And then if you infect them and you look in the different, you look at cells in the, I guess in the ear dermis, and we're looking in this ear after infection. Now that they're sharing blood supply, all of the T cells, eosinophils, neutrophils, and the monocyte-derived populations in the ear dermis are now of both, because they're sharing blood supply, they're of both, from both mice, but not the P4 population. If we look at the ear from this parabiont, the 45.2, all of the cells are 45.2 positive. So these tissue-resonant dermis macrophages, again, they are not replaced by blood monocyte-derived cells. So now we're going to compare the infection of the healing and non-healing strain and we're using RFP labeled parasites, and we recover all of the infected cells from the skin at day 2, day 5, and day 12 post-infection. And we find that at the early time points, well, so first of all, you'll note that all of the monocyte-derived populations, at least some cells are infected. They're phagocytes. If they find a parasite, they're going to take them up. The only difference in terms of the efficiency and uptake was the P4 dermis resonant population. Just like in vitro, bone marrow macrophages more efficiently took up the non-healing strain compared to the healing strain. It's not like they didn't take up the healing strain at all, but they took it up more efficiently. So this non-healing strain gets into all of these phagocytes, just more efficiently into the dermis resonant macrophages. And then if we look later in infection, so I think this is at 9 weeks and 12 weeks, and we recover the infected cells from the lesion. And we stain for iNOS, which is the critical inducible anosynthase that generates nitric oxide required for killing of these parasites inside of a macrophage. All of the monocyte-derived population, the P1s are inflammatory monocytes. These are new monocytes recruited to a lesion. These are these monocyte-derived macrophages and dendritic cells. These are infected, and they make a lot of iNOS. These infected P4 cells, despite the fact this is a lesion that has huge numbers of CD4 and CD8 cells making interferon gamma, it cannot make iNOS. But it does make a lot of arginase and realm alpha. These are markers of M2 functionality. These are critical upregulated markers for alternatively activated macrophages. Okay. So these P4 dermal macrophages maintain M2 functionality during infection. And again, that's despite the fact that this is, in terms of the T cell response, is primarily a Th1 immune environment. And then what he found was that this non-healing infection with this strain, this African strain, was mannose receptor dependent. So if we infected mannose receptor deficient animals, they developed a chronic lesion, but it was controlled similar to when we infected wild type mice with the normally healing strain from the Middle East. So these dermis resonant macrophages, at least expressing mannose receptor, were required for the evolution of this non-healing lesion. And then we got an antibody from Amgen. So it turns out these alternatively activated mannose receptor high macrophages are often found in tumors, in solid tumors. And when they're found in solid tumors, it's a bad prognosis for tumor development and tumor metastasis. So companies have developed reagents to try and get rid of these tumor-associated macrophages. And and some companies have actually targeted the mannose receptor, which is upregulated. Amgen targeted MCSF receptor because these tumor-associated macrophages and other tissue-resonant macrophages, remember, they're embryonically seated, but they're still dividing very slowly. They're self-maintained. And that turnover requires a macrophage colony stimulating factor signaling through the MCSF1 receptor. So antibodies against that receptor, if you treat for many weeks, you can deplete the dermis resonant macrophages in these mice. And when we do that and infect them, they no longer develop non-healing lesions. Again, they develop lesions that are pretty chronic but are controlled. So the depletion of targeting just that dermis resonant macrophage population it beautifully converts them to a more healing, resistant phenotype. And that population, again, this is a Th1 polarized mice. If you look at the level of T cells, you really can't find many T cells making IL-4 or IL-10. But if you look in IL-4 genetically deficient or IL-10 knockout mice, and particularly IL-4 and IL-10 double knockout mice, this population, this dermis resonant population, completely disappears. So even though it's a Th1 immune environment during infection, they still need IL-4 and IL-10 from some source in order to maintain their functionality. And so it's now known that IL-4 is in fact the key mediator of the localized turnover, self-turnover, self-renewal ability of tissue-resident macrophages. So IL-4 induces local proliferation of dermis-resident macrophages during infection. So what's the source of this IL-4? Again, it's very hard to see this. Certainly at the level of T cells, we can't see T cells making IL-4 very effectively unless we use some special mice, which I'll show you. But we were struck by the fact that in these lesions, we see T cells coming in, so they may be making small amounts of IL-4, or eosinophils. We see a lot of eosinophils coming into these inflammatory lesions. So I'm going to talk a bit about eosinophils and T cells as possible sources of IL-4. So IL-4 production, impossible to detect by intracellular staining, but some reporter mice for IL-4 production, which is a very sensitive way to look at IL-4 producing cells. They actively transcribe IL-4 message, and then these mice, these KN2 mice, these are mice that can be used to detect active cells that are actively producing IL-4 protein and not just transcribing IL-4. So in these lesions, we see these IL-4 positive cells. If we look at these KN2 mice that are actually making protein, it's both T cells and a lot of eosinophils that are secreting IL-4. So we're going to look at some intravital imaging again, focusing now on eosinophils. Well, in this imaging, oh no, we're just looking at, again, the perivascular location of these dermis resonant macrophages. This is a vessel. This is an amastogote. This was interesting to look at. This amastogote in this vessel, we can see this dermis resonant macrophages pulling in from the vessel this amastogote. So at least in this case, this seems to be a unless these amastogotes are inside of a neutrophil, which is possible, this seems to be a dermis resonant macrophages becoming directly infected. Okay, strategy for eosinophil visualization. So we use this manocept to stain dermis resonant macrophages. And then we have an antibody against this molecule Siglec-F, which will deplete, will stain for eosinophils. And then we're going to inject fluorescent parasites, in this case using a needle, not flies. So this is confocal microscopy, just a tissue section of the ear. This is a naive ear. And you do see a few eosinophils, and they're associated with these dermis resonant macrophages, even in steady state, but there's not so many eosinophils. But now, 12 days after a needle challenge, so the red are the Leishmania infected cells, which we think are inside of the dermis resonant macrophages. Now, in these inflammatory sites, we see quite a few more eosinophils, and they seem tightly associated with Dermis resonant macrophages in these tissues. So almost all of the Dermis resonant macrophages, when they're recruited to these inflammatory sites, they have this very tight association, intimate association with Dermis resonant macrophages. Okay, now we're going to do some dynamic imaging with another reporter mouse that we obtained. These are Eocre by Rosastop GFP mice. So because these are Eocre mice, the bright cells you see, these green cells that you see, no, sorry, the red cells you see are going to be eosinophils. So it's in these mice, only the red cells are eosinophils. And this is naive skin, so non-infected. So you do see eosinophils crawling around, and these white cells are the Dermis resonant macrophages. So in steady state, you do see eosinophils crawling around the tissue, not so many. I guess this is a higher resolution. Again, naive skin, steady state. There's not going on. Again, it's impressive, this staining. You can see how the perivascular positioning of the Dermis resonant macrophages. The vast majority of them you see lining the vessel. Again, I guess a higher resolution, again, naive skin, but you can see these eosinophils, even in steady state conditions, they're tightly associated, intimately associated with the Dermis resonant macrophages around these vessels. Okay, now we're doing infection. This is 1 hour post-infection. So now eosinophils are a bit more active. Still, the majority of them seem to be associated with the Dermis resonant macrophages. But not a lot of movement. Okay, now we're just 1 day post-infection. Now a huge number of eosinophils have been recruited into these infection sites. And almost all of them are associated with, you don't see a lot of movement. They've been attracted directly to these macrophages in the site. It's difficult to know from this slide whether which macrophages are infected or not. But again, it's impressive the number of eosinophils that have come in associating with these, and they're not moving about. And they're not dying the way neutrophils would come in and die and be taken up. Okay. So what happens when we deplete neutrophils with when we deplete eosinophils with this anti-Siglic F antibody? So this is controlled, treated mice. I don't know how many days after infection this is, but this is treating the mice with this antibody, and it very efficiently depletes these eosinophils from the skin. And doesn't affect the other populations. But if we now, doesn't directly affect, but if we now look at the numbers of P1, P2, P3. These are the monocyte-derived cells. It's only the Dermis resonant macrophages, the P4 cells, that are now halved in number, just because we've gotten rid of eosinophils. So they are contributing, at least in part, to the maintenance of the Dermis resonant macrophage population during infection. And we think it's because they're delivering IL-4. Now, IL-5 is a major cytokine to recruit and activate neutrophils. When in IL-5 deficient animals, the number of Dermis resonant macrophages is also depleted. And we think that's because the number of eosinophils has now gone way down in these IL-5 deficient mice. And then if we just do the infection, we see this at least a partial phenotype in terms of lesion development in the IL-4 knockout and IL-5 knockout animals. So we conclude that we have, first of all, defined this Dermis resonant macrophage population that's present in steady state conditions, and they express these M2 alternately activated macrophage markers, and they maintain their M2-like phenotype even in this strong Th1 immune environment. They turn over, they're embryonic derived, but they do undergo this slow division turnover, which is IL-4 dependent. So they locally proliferate. They're not replaced by monocyte-derived cells. And eosinophils may be a key source of IL-4 to maintain dermal resonant macrophages. They're tightly associated with, the eosinophils are tightly associated with the macrophages. Depletion of eosinophils, IL-4 and IL-5, results in decreased numbers of Dermis resonant macrophages during infection. So more generally, these Dermis resonant macrophages, as well as we think other tissue resonant macrophages, they serve as favorable replicative niches for Leishmania major. And their preferential infection by some strains of L major explains the severity of disease. But for all strains, even the ones that heal, because we saw even the healing strains infected these Dermis resonant macrophages, just not preferentially. And that probably explains why there's no sterile immunity, why there's persisting infection even when these infections are controlled. Because these cells really can't be activated for killing. That's not why they're there. They maintain this M2 functionality for a lot of reasons, but the biology of these cells and their intimate association with eosinophils is part of their normal biology. So their persisting infection may explain the failure to achieve sterile cure even in healing strains. If we think about visceral leishmaniasis, same thing. We have persisting infection after healing. We have persisting infection in the skin, which can be reactivated to cause PKDL. Are the persisting infections due to parasites inside of these tissue resonant macrophages, which are, again, their biology is so different. They are embryonic derived. They promote a reparative function, a homeostatic function for capturing dying cells, for repairing tissue, for resolving inflammation. But they are a beautiful and suitable environment for intracellular pathogens to if not grow, at least not be killed. So we think these are an important cell in all of these tissues to look at that might explain persisting infections in leishmaniasis and persisting infections in other infections like tuberculosis. Why there's rarely, if ever, sterile immunity in these kinds of infections. And I think with that, we'll end there and take whatever questions you might have. Okay. Questions? Yeah.
So, in that EO3 mouse, what is the exact location of the 3 gene inserted? The gene is inserted?
So it's, so that's a good question. I don't know what they've targeted that is eosinophil specific. So I should look at that. It may be the receptor for eotaxin, which is the eosinophil chemotactic factor, but that's a good question. But these mice have only recently been developed, and that imaging, that live cell imaging of eosinophils in the skin, I think we were the first ones to actually look at eosinophil behavior in the skin. That hadn't been looked at before. So, and certainly this interesting and intimate association with macrophages in the skin had not been seen. So, it's an important point that when, as beautiful as this intravital imaging is, you can only see what your system allows you to see. So you need certain kinds of reporter mice. So for years we had done imaging because we had the lysozyme M mice that allowed us to see neutrophils. But we didn't have any way to stain Dermis macrophages, so they were excluded from our understanding of the biology of these infections just because we couldn't see them. And we weren't looking at eosinophils before. So just keep that in mind. You're not seeing everything that's going on. These systems are being developed more fully so that you can look at multicellular parameters. But these two photon microscopes they're very expensive, and their physics is constantly being upgraded so it can penetrate into deeper tissue. But you know, they're capturing images over many hours. That's how these images are captured. But really important to remember, you're only going to see what your tools allow you to see. And we are almost certainly missing other critical events. But at least the things you are seeing, you believe because you're seeing it. These are not, there's no sense of artifact to these real-time intravital images that you're capturing. They're at least a part of what's really going on. Okay. Yes?
The area, the part where the eosinophils are attracted, was there any study where they have measured the chemokine ligands in that area which are specifically attracted to the eosinophils, or what is the source of
yeah. So the data I didn't show you is that, in fact, that attraction, and I should have showed that data. So remember in vitro when we put IL-4 and IL-10 on bone marrow cells, and they upregulated mannose receptors so impressively. So if we take those bone marrow cells and treat them with IL-4 and 10 and look at the chemokines they produce one chemokine, and that's eotaxin. I should have showed that data. And it wasn't IL-4 alone, IL-13 alone, IL-4 and 13. It had to be IL-4 plus IL-10. And then in vivo, in the IL-4, IL-10 double knockout mice, remember those Dermis macrophages, they completely disappeared. It's because we think they really do require these eosinophils to be maintained. And that condition, IL-4 plus 10, even though there's so little of it in the skin, and that's another sort of important lesson. These assays we use to study cytokine production, it doesn't take into account that very low levels of production, if they're produced by cells in such tight association with their target cells, can have profoundly important effects. So you may not, your assay systems may not be able to detect it. That doesn't mean those cytokines are not having a really profoundly important effect on the biology of these infections. So, yeah, I mean, that's another important, somewhat discouraging lesson that we miss a lot because of just the kinds of assays that we do. And it's not surprising when we look at cytokine production in these conventionally resistant B6 mice and we see so many T cells making interferon gamma, we think, well, that's all that's going on. Because this is such a classical Th1 mouse, this L major infected B6 mouse, and yet they're not healing infection because of events that are very difficult to see. But if you look carefully and locally, there are cellular interactions which have these beautiful effects. Yeah, other.
On the fact, I think, that if some free pathogens are present in the bloodstream and they are interacting with bone marrow-derived monocytes, and can they change the chemokine receptors on the host cells so the same cells can go in the tissue and reside there?
Yeah, that's a nice question. Well, so I don't know the answer to that. Yeah.
Were the eosinophils depleted in the peripheral blood also?
Yes, these treatments would have depleted in the peripheral blood as well, but these are transient depletions. So there are eosinophil knockout mice, and I didn't show that data. All right, well, consider some issues, questions, concerns. So for after lunch, and I think we'll have figured out what review papers to be assigning to each of the different groups at that time. So we're meeting back here at what time?
We have to also come back after lunch. 1:30 will be okay?
So 1:30.